Hello and welcome to Caltech's 2022 3-minute thesis competition. My name is Kara Watley and I'm the University Librarian at Caltech. And Caltech Library is delighted to partner with the Graduate Studies Office, Public Programming, and Theater Arts at Caltech for this event. We're delighted to have all of you join us here in person, and we're delighted to have all of you joining us via live stream. Before we begin our program tonight, I want to acknowledge and offer our respect to past and present Gabrielino Tongva people and their unceded ancestral lands, including the LA Basin upon which Caltech sits today. We invite you to honor the Gabrielino Tongva with us and to acknowledge the Native, First Nations, and Indigenous peoples who have stewarded the lands for which you and your hometown may occupy. Now let me say a little bit about the 3-minute thesis competition. It was originally conceived by the University of Queensland, and it challenges Caltech graduate students to explain their research in an engaging and clear 3-minute talk intended for a non-specialist audience. Given Caltech's prominence in science and engineering communities, we feel it's important to give Caltech graduate students the opportunity to practice communicating their science in ways that will both resonate and inspire. This year's competition received 39 entries, the most that we've ever had for a Caltech 3MT, and we chose 14 finalists to present their research today. I would like to say thank you very much to our preliminary judges, George Porter and Kathy Johnson from the library, and Kara Steemans and Mary Herrera from Public Programming for all of their work. So this evening, our 14 finalists will be competing for 4 prizes. Prizes made possible by the generous support of our sponsors for this evening's event. Our sponsors this evening are EBSCO, Elsevier, American Physical Society, IOP Publishing, Science of Synthesis, Tema Chemistry, Wiley, and Springer Nature. Through their generosity, our finalists will be vying for a $3,000 first prize sponsored by EBSCO, a $2,000 second prize sponsored by Elsevier, a $1,000 third prize sponsored by Wiley, and a $500 People's Choice Award chosen by those in the audience tonight here in the auditorium and also online. Representatives from each of our sponsor companies are attending via live stream this evening, and I would like to thank them for all of their support for our Caltech graduate students and for science communication. It's now my pleasure to be able to introduce you to our Master of Ceremonies for this evening's event, Brian Brophy. Brian is a 25-year veteran actor in the film, TV, and theater industry, and he's also the director of Caltech Theater. Brian is also a Fulbright Scholar in India, a George Soros Award winner in Bosnia, and a story coach with the Jet Propulsion Laboratory. Brian?
Thanks, Kara. I would now like to introduce our judges who will select the first, second, and third place winners. You, the audience, will vote for the People's Choice Award. Our judges are Professor Doug Reese, our beloved Dr. Doug Reese. Professor Reese is Caltech's Roscoe Gilkey Dickinson Professor of Chemistry and an investigator at the Howard Hughes Medical Institute. In his years of service to Caltech, Doug has been executive officer for chemistry and for biochemistry and for molecular biophysics, and most recently served as dean of the graduate studies. Doug's research interests on the structural biology of metalloproteins and membrane proteins and have been propelled by the graduate students and postdocs in his group. Thanks, Doug. Second is Dean Daryl Peterson. Daryl is the Associate Dean of Graduate Studies at Caltech. He received a BA in Communication and an Master's in Education in Counseling Psychology, both from James Madison University in Harrisonburg, Virginia, and a PhD in Educational Leadership from the Iowa State University of Science and Technology in Ames, Iowa. Daryl has over 25 years of experience in higher education and student affairs and has worked with both undergraduate and graduate students at a variety of institutions from coast to coast. Here at Caltech, Dean Peterson coordinates new student orientation, provides support for graduate students in crisis, and serves as a member of the CARE team. Thank you, Daryl. Third, we have Dr. Donna Wroblewski. Donna received her SB degree in chemical engineering from MIT and her MS and PhD degrees in polymer science and engineering from the University of Massachusetts at Amherst. She worked for 4 years as a junior scientist at Solutia Inc., working on improving the properties of glass polymer laminates before being recruited for a tenure-track librarian at the University of Florida, where she was responsible for reference, instruction, and collection development for the departments of chemistry, chemical engineering, material science, and engineering, and physics. At Caltech, Donna currently serves as the Chemistry and Chemical Engineering Librarian and the head of the Research Services Department at the Caltech Library. She is also the chair of the Division of Chemical Information of the American Chemical Society. Wow. Number 4, Dr. Susanna Hall. Dr. Hall is a teaching professor of writing and director of the Hickson Writing Center at Caltech. Dr. Hall holds a PhD in English from the University of California, Irvine. Her current research focuses on academic scientific communication. At Caltech, Dr. Hall teaches a variety of writing classes and courses, including communicating science to non-experts. Much needed. Number 5, Dr. Robin Javier. And there she is over there. Dr. Javier is a STEM communication specialist and lecturer in the Division of Engineering and Applied Science. She has a PhD in neuroscience, a master's degree in biomedical engineering, and more than 15 years of experience in science and engineering communication. Here at Caltech, she teaches courses and workshops focused on presentation skills for undergraduates, graduate students, postdocs, and the broader campus community. In her free time, she is also a freelance writer, illustrator, and creative scientific storytelling consultant. Thank you. Good, that's it. So, what are the criteria for judging the presentations, right? That's what you're wondering. Well, number 1, did the speaker clearly convey their thesis and the significance of the results to the audience, right? That's number 1. Did the speaker clearly convey their thesis and the significance of the results to the audience? Number 2, did the presentation provide adequate background to the research question? That's number 2. 3, did the speaker have sufficient stage presence, eye contact, and vocal range, maintain a steady pace, and have a confident stance? Number 4, did the oration make you want to know more? So, now we're going to get right to the presenters. Yes? All righty. So excited for this moment here. So, here we are. Emily de Jong is in the Division of Engineering and Applied Science. Tapio Schneider is her lab in the Climate Dynamics Group. John Seinfeld is the co-advisor. Thank you.
You've probably heard a range of predictions about global warming. How much will temperatures increase in the next 50 years? How much will sea levels rise? Predicting future climate is a difficult and highly uncertain task, But what makes climate so much more difficult to predict than, say, local weather? Well, for one, climate models have to forecast on much longer timescales and over the entire planet, rather than just your neighborhood. It's really challenging to model all of the complex and chaotic processes of Earth's atmosphere in a way that's simple enough to run on a supercomputer. The focus of my research is one of the most challenging aspects for these models to capture: clouds. Clouds are crucial to the Earth's climate. Some clouds reflect incoming sunlight and cool the Earth. Other clouds trap heat and warm the Earth. In order to predict future climate, we need to understand how clouds react when humans pump CO2 and aerosols into the atmosphere. Clouds themselves are made up of trillions and trillions of tiny water droplets suspended in the air. These droplets can grow and shrink, they can collide and coalesce with each other, they can freeze and become ice, and sometimes they become so large and heavy that they fall out of the sky as rain. Notice that all of these processes occur at micron-sized droplet scales, which we definitely cannot represent in a global climate model. Instead, we abstract all of these tiny droplets down to just 1 or 2 numbers. For instance, the amount of liquid water in the cloud, or how well it reflects light. In doing so, we lose a lot of droplet scale physics, like influences from aerosols, and this contributes to the huge uncertainties we have about future climate. While we cannot model cloud processes directly, we can do so more smartly. In my research, I improved the way that we model cloud physics by developing new mathematical techniques that take advantage of our ever-increasing computational resources. The more we know about a population of cloud droplets, the better we can predict how those droplets will evolve, and the more confidence we'll have in the clouds that traverse the skies of our climate model. I developed computational methods to keep track of more detailed information about clouds, like the number of droplets of different sizes. With this additional detail, we can incorporate more droplet scale physics, like influences from aerosols. With these improved models, we can then better predict trends like drought, rainfall, and perhaps most importantly, how and where clouds redirect energy from the sun. My microscale research in cloud physics modeling can one day reduce uncertainties about global scale climate change. Thank you.
Thank you. That was Emily DeYoung, a third-year graduate student in mechanical engineering at Caltech, where she researches atmospheric microphysics modeling with Professor Tapio Schneider and Professor John Seinfeld. After obtaining her BSc in chemical engineering from Princeton University in 2019, Emily found her way into the atmospheric sciences through her interest in fluid dynamics. When she is not modeling clouds and aerosols, Emily can be found doing observational field work, that is, marveling at the sky while hiking, climbing, or riding her bike. Thanks. Next we have Christopher Kenseth from CCE, John Seinfeld Lab with Paul Winberg and Brian Stoltz, pulling molecules out of thick air.
Living in Los Angeles, you cannot help but notice the air. On most days, you can see the air, but not the nearby San Gabriel Mountains. By and large, airborne particles, collectively termed atmospheric aerosol, are responsible for this view-obscuring pollution that reduces our air quality, impacts our climate, and adversely affects our health. Aerosol particles are ubiquitous in the atmosphere and originate from a wide variety of human and natural sources. For example, wildfires, desert dust, and fossil fuel combustion. These particles are small, with diameters ranging in size from a few nanometers, smaller than viruses, to tens of micrometers, about the width of human hair. Aerosol particles exert large but highly uncertain effects on Earth's climate, directly by scattering incoming sunlight back to space, and indirectly by seeding cloud formation and altering their reflectivity in lifetime. In fact, without aerosol particles, there would be no clouds in our atmosphere. As many of us have experienced during our time at Caltech, aerosol particles also adversely impact air quality and, by extension, human health. Exposure to ambient aerosol is associated with increased risk of respiratory disease and is estimated to cause up to 9 million premature deaths per year globally. Now, aerosol particles can either be directly emitted into the atmosphere from a source, termed primary aerosol, or formed in the atmosphere through oxidation of gaseous emissions, termed secondary aerosol. In Shanghai, for example, most of the urban smog arises via direct emission of aerosol particles from coal-fired power plants surrounding the city. By contrast, the smog in Los Angeles is mainly formed from oxidation of emissions from human activity. For example, uncombusted gasoline vapors and tailpipe exhaust. Similarly, the smokiness of the Great Smoky Mountains results almost entirely from the oxidation of gaseous compounds emitted from plants and trees. That said, globally, most aerosol particles are formed in the atmosphere through oxidation of organic gases, those that contain carbon, and are hence termed secondary organic aerosol, or SOA. SOA, unfortunately, is highly variable and chemically complex. Human and natural sources emit thousands of organic gases that could form SOA, and even when formed from the same precursor, differences in environmental conditions, for example, temperature, humidity, and available sunlight, impact the oxidation chemistry and result in compositionally distinct SOA. Owing to this chemical complexity, characterization of the formation, properties, and fate of SOA in the atmosphere represents a formidable challenge, leading to significant uncertainty in assessment of its environmental and health impacts. This is where my research comes in. Rather than make measurements of SOA amid the immense complexity of the real atmosphere and struggle to decouple all of the potential variables impacting its formation and properties, I studied the composition of SOA formed in a massive 24,000-liter Teflon chamber under highly controlled environmental conditions. I focus on SOA formation from the most abundantly emitted and thus important organic gas precursors, such as pinene, the compound responsible for the smell of pine trees, and investigate how the molecular composition of SOA formed from these precursors varies as a function of temperature, humidity, and other environmental factors using a suite of advanced analytical instruments. My name is Chris Kenseth. I'm an atmospheric aerosol chemist, and in this way I pull molecules out of thick air.
Thank you. That was Christopher Kenseth, a PhD candidate in the Division of Chemistry and Chemical Engineering, jointly advised by Professors John Seinfeld, Paul Winberg, Brian Stoltz. He received a BS in chemistry from the University of Vermont in 2015. He is the recipient of the 2021 Herbert Newby McCoy Award, which recognizes the most outstanding achievements in research by a chemistry and chemical engineering graduate student. This July, he will begin an NSF Atmospheric and Geospace Sciences postdoctoral research fellowship in the Department of Atmospheric Sciences at the University of Washington with Professor Joel Thornton, where he will combine organic synthesis, laboratory experiments, atmospheric modeling, and field measurements to study the chemistry of terpene organic nitrates. Thank you. Next up, Yiming Li from HSS and the lab of Michael Alvarez. Universal mail ballot delivery boosts turnout. The causal effects of sending mail ballots to all registered voters.
Good afternoon, everyone. Today I'm presenting my research, Universal Mail Ballot Delivery Boosts Turnout, the causal effect of sending mail ballots to registered voters, joint work with Mike Alvarez. While the practice of sending mail ballots to registered voters was gaining popularity before the 2020 general elections, it was suddenly used in a larger number of states due to the COVID-19 pandemic. While some states, including California, have decided to continue universal mail ballot delivery for future statewide elections, some other states have moved to restrict voters' access to voting by mail. It's therefore crucial to understand the effect of this policy. In this research, we are going to take advantage of a unique situation where for the 2020 primary elections, in addition to permanent absentee voters, Los Angeles County was required by state law to mail ballots to registered voters living in federal congressional districts, as well as state legislative districts that contain areas in both Los Angeles County and Orange County. Therefore, this generates a quasi-natural experiment where voters living in universal mail ballot districts, color-coded orange on this slide, will automatically receive a ballot, whereas the control voters are those living in other districts, and they would only receive a mail ballot if they had requested one. Looking at the main results presented in the middle of the slide, we see that voter turnout increased from 2018 to 2020 for both the control group and the treatment group, but the increase was larger by about 3 percentage points for the treatment group, indicating a 3% boost in turnout due to the policy. Breaking down the effect by party registration, we see that voter turnout increased due to the policy for voters regardless of their party registration, but effect was larger for registered partisan voters compared to those without the party affiliation. We also find the effect to be about half percentage point larger for racial minority voters compared to non-Hispanic white voters. In order to control for further control for potential differences in terms of political competitiveness across congressional districts, we also look at the voters residing in California's 32nd and 40th congressional districts, where part of this congressional district were subject to universal vote by mail. We see some heterogeneity, but the main takeaway remained the same. To conclude, we find that universal mail ballot delivery increased turnout by about 3 percentage points for voters who do not otherwise receive a mail ballot, and the effect was larger for richer partisan voters compared to those without party affiliation. Our results suggest that expanding voting by mail to voters increased turnout while restricting their access to voting by mail could lead to voter disenfranchisement. Thank you.
Thank you. That was Yiming Li, a PhD candidate at the Division of the Humanities and Social Sciences at Caltech. His research focuses on elections and statistical methods. He holds a Master of Science degree in econometrics and mathematical economics with distinction from London School of Economics and Political Science, and is the co-author of the book, Securing American Elections, How Data-Driven Election Monitoring Can Improve Our Democracy. Thank you. Our next presenter is Max Saccone, a CCE, Julia Greer's lab, harnessing combustion to 3D print metals.
Creating physical objects is a fundamental human endeavor. From tools to toys to sculptures, acts of creation allow us to find meaning and improve our quality of life. We've even named entire eras of our history after common materials, from the Stone Age to the Bronze Age to the Iron Age and beyond. Today, we find ourselves on the dizzying crest of rapid technological development. We face unprecedented and existential challenges, such as anthropogenic climate change, but we also have more tools and materials than ever before to tackle these challenges. One of the most recent advances in our ability to engineer materials is the process of 3D printing, which allows us to create complex, computer-designed structures one layer at a time. Imagine a sculptor who chips away at a block of marble to remove material where they don't want it. 3D printing flips this around by only adding material where we do want it. Often, this allows us to make things more quickly and with less waste. Or we can flex our imaginations to realize previously impossible designs. A remaining challenge in the field is how to 3D print metals with very small features. The best existing methods use a laser to melt and solidify metal powders. However, this doesn't work well for some materials like copper, which transmit heat so well that even if you have a finely focused laser, the heat spreads out and melts too much powder, reducing the resolution. To overcome this challenge, we had to invent a new way to 3D print metals, and we knew that we couldn't rely on heat to define the part shape. Instead, we use ultraviolet light to join together molecular building blocks called monomers into a loose network called a hydrogel. We then convert this hydrogel into metal by infusing precursors and then taking advantage of chemical reactions like combustion and reduction. With this technique, we can 3D print copper with feature sizes about half the width of a human hair, much smaller than previously possible. And by studying these improbable transformations, we learned how to apply the paradigm of infusion and reaction to make lots of new materials, from battery electrodes to multi-component alloys, all with exquisite resolution. We hope to use these materials to solve societal problems, from making more robust batteries to lighter structural materials. We also hope that other researchers will take advantage of the tools we've developed to make their own new materials and follow their own journeys of innovation and discovery. Beyond 3D printing, each one of us has tools we can use to make the world a better place. What are your tools, and what will you make? Thank you.
Thank you. That was Max Saccone, a PhD student in chemical engineering, working with Professor Julia Greer. He graduated from Dartmouth College in 2017 with an AB and BE in engineering sciences and a chemistry minor. Max's research at Caltech focuses on developing additive manufacturing techniques to enable the design of 3D architected battery materials, as well as other functional materials such as metals and alloys. Max is an avid rock climber and trail runner and finds meaning in environmental stewardship in many forms, from trail maintenance to sustainability-focused research. Thank you. Next up is Christian Eldarn, a BBE, Lior Pachter's group. how a tree can save your life.
CMS, actually. Yeah. Any viral outbreak takes on the shape of a tree, and not the type of tree you would find in your backyard, but a family tree of infections stemming from the first person infected in the outbreak. And as the virus transmits from person to person, it accumulates new mutations. If we know the sequence of the viral genome in each infected individual, we can use statistical methods to piece together who infected whom. So we can essentially recover the likeliest tree of transmissions. Now, there are two main things that affect the infectiousness of a virus. It is the physiology of the virus itself, and it is the behavior of its hosts. And this tree tells us something about human behavior in a pandemic setting. Namely, if you infect a lot of people, you generate a lot of branches in the tree, whereas if you infect no one, you are essentially a leaf. Now, if we have the tree in front of us, we can identify individuals that are more infectious than others, and we can identify common characteristics between those people. And then when it comes time to vaccinate the population, we can target people that have these common characteristics. So when COVID-19 spread its roots around the world, I was recruited to be part of the COVID-19 task force in Iceland. And in Iceland, we sequenced the viral genome of every single diagnosed case. We used these data to reconstruct the maximum likelihood transmission tree of a single very large outbreak of COVID-19. We found that people who were outside of quarantine at the time of diagnosis were 89% more infectious than people who were in quarantine at the time of diagnosis. And this isn't news to us. We know that quarantine works. We've been doing it for thousands of years. But it is incredibly difficult to quantify exactly how effective quarantine is, because that requires data on the individual level, like we have in the transmission tree, rather than some summary statistic about the whole, which is what we usually have. We also found that people of working age were 47% more infectious than children and people of retirement age. And just the fact that different groups of people are not equally infectious suggests to us that we might be able to strategically distribute vaccines in order to maximize the efficacy of every single dose. So we simulated different vaccination strategies on our transmission tree by strategically selecting people to be immune and then pruning all the branches stemming from them. We showed that vaccinating a population uniformly at random is substantially better than vaccinating a population in order of descending age. And this is the fruit that our research ultimately bore. We presented these results to the healthcare authorities of Iceland, who changed their vaccine rollout strategy from the descending approach to a random approach in order to attain a higher level of protection sooner. And that is how randomly pruning a figurative tree can literally save your life. Thank you.
Thank you, Christian. An Icelandic musician, artist, and PhD candidate in computing and mathematical sciences, yes, under the tutelage of Leor Pachter. While his scientific endeavors mostly involve developing new graph-based methods for solving open problems in computational biology, they sometimes bleed into his music and art, resulting in the creation of never-before-seen instruments and complex generative algorithms of artistic creation. Thank you. Next up is Skylar Ware in CCE, Kim C's group, Sports Drinks for Batteries and Beyond.
Energy from renewable sources like wind and solar produces no greenhouse gases and is one of our best defenses against rising global temperatures. But those energy sources aren't constant. They come and go with the weather and the time of day. If we want to use solar power at night or wind power on a calm day, we need a way to store that energy for later. To do that, we need to develop new and better types of batteries. Most batteries have three main parts: the cathode, what most folks think of as the positive end, the anode, or the negative end, and an electrolyte solution, which sits between the two. Electrolytes are particles that carry a positive or negative charge, and they're usually dissolved in a liquid, just like dissolving table salt in water. Electrolytes help maintain balance in machines that store energy, like batteries or human bodies. In fact, the electrolytes in a battery aren't so different from the electrolytes in your favorite sports drink. Just like how the electrolytes in that sports drink move through your body to keep you hydrated and power your muscles, electrolytes can also move through a battery to keep electricity flowing and power a device. But while we know a lot about the different types of electrolytes that our bodies need and about how we use those electrolytes, we don't know as much about which electrolytes are best for the new batteries we're developing to meet our energy storage needs. So how do we find the best electrolytes for the job? Well, that's where I come in. I study how different electrolytes interact with other parts of the battery, specifically with the anode. In most cases, the liquid electrolyte solution reacts with the solid anode to form a surface film, aptly named the solid electrolyte interface, or SEI for short. In the batteries we use now, the ones in our phones and laptops, the SEI lets electrolytes move in and out of the anode when we charge and discharge the battery. But in the new types of batteries we're trying to build, it doesn't always work out that way. Some SEIs block or divert electrolytes from getting where they need to go to keep our battery working. And it's not always clear why those SEIs don't work. My research aims to understand the relationship between the electrolytes we use in a battery, the SEI they form on the anode, and the battery's performance. So far, I've developed very concentrated electrolyte solutions that can be used in batteries with lithium metal anodes. I've shown that in those systems, electrolytes that contain lots of fluoride perform the best. Fluoride reacts with the clean lithium metal surface and forms a more stable, less reactive SEI. Once that happens, it prevents the rest of the electrolyte solution from reacting in ways that are harder to control. My work goes beyond batteries too. Using the lessons we've learned from developing battery electrolytes, I've worked with synthetic chemists who use electricity to make medicines and studied the electrolytes and SEIs that make those syntheses more efficient. So understanding how electrolytes interact with everything around them will help us use electricity more efficiently in everything from all-electric cars and airplanes, to running our cities on renewable energy, to space exploration and beyond. Batteries can do it all. They just need the right sports drink. Thank you.
That was Skylar Ware, a fourth-year graduate student in the Division of Chemistry and Chemical Engineering, advised by Professor Kim See. Her thesis research focuses on developing new electrolytes for next-generation energy storage systems and electrocatalysis. She is originally from Ohio and earned her bachelor's degree in chemistry from The Ohio State University in 2018. Outside of the lab, she enjoys hiking, reading, and playing tabletop RPGs. Next up is Himani Kalucha from GPS, Michael Brown, Planetary Ice Chemistry Lab, Detecting Life on Europa.
Hi everyone, my name's Himani and I'm a second-year graduate student in the geobiology program. Scientists think that one of the most promising places to find life in the solar system is Europa, a moon of Jupiter. And Europa is a really special place because one, it has liquid water, and two, it is one of 3 worlds in the solar system amongst hundreds where the ocean is in touch with the rock, the other 2 being Enceladus and Earth. And water-rock reactions are really critical to life on Earth. They produce a host of interesting chemistry, and water itself is great at dissolving and transporting nutrients. So if life exists on Europa, it's probably down in these hydrothermal vents or at the ocean ice interface. And you could imagine it coming up through the ocean currents, through the ice, and the bacteria being deposited on the surface. Once that happens, they're hit by charged particle radiation, which is probably going to kill them, but that's okay. We can still work with that. I'm interested in studying how this radiation is going to chemically change the bacteria. And I do this by taking bacteria from real hydrothermal vents in Earth and putting them into ice mixtures. And then this mixture goes into this vacuum chamber you see on the right, which goes down to Europa's surface temperature and pressure, a cold 78 Kelvin. I then irradiate them with my electron gun, and after the end of the experiment, I have what the bacteria look like after years on the surface of Europa. Through these windows, I can take spectra, which produce unique peaks, as you can see down here, corresponding to organic molecules that are present in the mixture. So once I have all this data, there's a mission going out called Europa Clipper that's going to orbit the surface of Europa, and when we get data back from it in 2030, we can compare it to this library of spectra that I've been creating. And this data is critical to distinguish, one, if the organic molecules that we might find are actually coming from inside the ocean or just being deposited by impacts, because meteorites actually carry a whole host of organics. 2, if the organic molecules are actually made by life or not. Because, for example, the solar system can make certain amino acids entirely without the help of life. And 3, if the organic molecule is made by life, how long ago was it alive? So while it's a dream to have a very hot big nuke that melts through 20 kilometers of ice and reaches the ocean one day, that's pretty far off from current missions. And my project represents a promising life detection method for the near future using existing mission capabilities. Thank you.
Thank you. That was Himani Kalucha, G2 in the geobiology option, co-advised by Woody Fisher and Mike Brown. She works on biosignature detection in the context of the Perseverance rover and Mars analog environments, as well as icy moon surfaces. She has an undergraduate degree in aerospace engineering. Next up is Sean Pike, Division PMA, in the lab of Fiona Harrison, New Star Group. Life after death, how the remains of massive stars light up the X-ray sky.
When a massive star depletes its thermonuclear fuel, it undergoes a violent explosion known as a supernova. This is often considered the death of the star, the end of its luminous life cycle. But depending on how things go, the star's iron core may be crushed into an ultra-dense soup of elementary particles known as a neutron star, or even wilder, a black hole, a region of time and space where where gravity is so strong that light can't even escape. When these stellar leftovers find themselves orbiting a living star like our sun, something interesting happens. If the two objects are close enough to one another, then material may be lifted off of the surface of the companion star and fall into orbit around the neutron star or black hole. Soon it forms what we call an accretion disk as it spirals inwards due to an interplay of gravity and internal friction. As it does so, it heats up, reaching temperatures in the tens of millions of Kelvin, thousands of times hotter than the surface of our sun. This hot material emits a huge amount of high energy photons, which we see as X-rays. We therefore call these systems X-ray binaries, and we need specialized space-based telescopes to view and to study their physical properties. At Caltech, I've primarily focused on using one of these observatories, the Nuclear Spectroscopic Telescope Array, or NuSTAR for short. NuSTAR takes images of the X-ray sky while also measuring the energy and the arrival time of each individual photon that it sees, making it an ideal instrument for studying the array of phenomena that we see from X-ray binaries. For example, when a neutron star accretes, it may trigger runaway nuclear burning on its surface, resulting in an impressive but short-lived flash of light as its entire surface is enveloped in flames. I analyzed NuSTAR observations which caught not one, but two of these explosions. And I looked at how material was lifted off of the surface of the star, and I analyzed spectral and timing properties in order to find the composition of the material, as well as the distance from the Earth to the neutron star. More recently, I used NuSTAR to perform rapid follow-up of a newly discovered X-ray binary. When it first appeared on the sky, we didn't know if it was a neutron star or a black hole, but we did know there was an accretion disk. By looking at how the emission from the disk was actually distorted by gravity, I was able to determine that it was in fact a black hole. By actually probing how the central object dragged space and time around it, I found that it was spinning way too fast to be a neutron star. If it were a neutron star, it would tear itself apart. So, studying the physics of these objects and how they accrete can tell us a lot about bigger questions in our universe, such as supermassive black holes in the centers of galaxies. Because X-ray binaries evolve on timescales that are reasonable for a graduate student to study, we can use them as analogs for these larger black holes, which evolve on much longer timescales. I like to think of X-ray binaries as, each X-ray binary as a sort of corner of the larger puzzle that our universe presents us, each telling a unique story about the hottest, densest, most energetic regions of space. Thank you.
Thank you. That was Sean Pike, a sixth-year physics graduate student in Fiona Harrison's group, where he works on a combination of X-ray astronomy and instrumentation. He received his bachelor's degree in physics from Brown University in 2015 when he completed a senior thesis measuring the apparent distortion of galaxy shapes by dark matter structures. Before starting grad school, he spent a year studying data science techniques at the Institute of Space and Astronautical Science in Sagamihara, Japan. This project introduced him to the field of X-ray astronomy, leading him to join the New Star group at Caltech. He plans to defend his thesis this summer, and he hopes to continue working in the field. Thanks, John. Next up is Ida Baymard from PMA, Andrew Howard's lab, Astronomy Department, Planets on a Galactic Scale.
I study exoplanets. These are planets outside the solar system, orbiting stars other than the sun. We discovered the first exoplanet only about 30 years ago, detected by a minuscule dipping of light that occurred again and again as the shadow of that alien world passed in front of its host star on its orbit. How many exoplanets do you think we've found now? Well, thanks to recent telescope surveys, just last month, that number passed 5,000. Like other exoplanet astronomers, I use specialized software to analyze telescope data of these numerous alien worlds. And what we found is surprising. Most exoplanets are totally unlike the planets in our own solar system. We found hot, gas giant planets orbiting super close to their host star, and planets with densities as light as that of cotton candy due to thick, dusty atmospheres. And the strangeness of these alien worlds means that the story of how planets form is much more complex than anything we previously imagined. So, how do we uncover the secrets of planet formation? Well, we can start with the stars that planets orbit. And that's because the characteristics of host stars, like how bright they are and what they're made of, encode clues on how their planets came to be. In my thesis, I look at the amounts of different elements in stars because each unique chemical fingerprint tells us something about the planetary system's formation. So far, I found host stars rich in rocky elements like iron. This is a sign that they ingested planetary material, maybe consuming their own planets in an act of engulfment. These systems tell us how much rocky material is typically eaten by stars and how much is left to form planet cores. And that reveals what kinds of planets eventually form. If there's lots of uneaten rocky material available, planet cores can build up fast and then accrete huge amounts of gas to form Jupiter-sized worlds. And if there's not much rocky stuff around, smaller Earth-like planets are more likely to form. I'm going to combine this work with another area of astronomy, galactic archaeology, which treats the chemistry of stars as artifacts that allow us to reconstruct the history of our galaxy, the Milky Way. By looking at rocky chemistry and galactic archaeology data sets, we can learn if small planets that may host Earth-like life are common throughout the galaxy, or worlds with different sizes, made of more exotic compositions, that could be home to something a little stranger. Then maybe we can answer the question, why are we here in this part of the galaxy and not somewhere else? So, just 30 years after we discovered the first exoplanet, it's now possible to investigate how planets form on a galactic scale. Who knows what discoveries of worlds beyond the solar system and what it means for our cosmic origins the next few years will hold. Thank you.
Thank you. That was Ida Baymard, a planetary science graduate student at Caltech, where she studies worlds beyond our solar system. She is also passionate about science outreach and volunteers as a K through second grade science teacher at underserved Pasadena public schools. When she's not doing science, she likes to hang out with her cat, interstellar medium Izzy for short. Next up is Philip Jahelka, EAS, Harry Atwater's lab, low-cost gallium arsenide solar cells.
All right, hi all, my name is Phil Jahelka. I'd like to tell you a little bit today about my work on low-cost gallium arsenide solar cells. I started working on solar cells because of the need to quickly decarbonize the world's economy in order to prevent further global warming. In particular, there are good reasons to want to limit global warming to 1.5 C. At 1.5 C of global warming, the Earth's coral reefs, which feed 500 million people, disappear. There are two fundamental bottlenecks that limit how quickly manufacturing capacity of solar cells can grow. The first is the capital intensity of turning sand into semiconductor-grade silicon, which is the material that is actually absorbing the light in a solar cell. The second are the very limited profit margins of solar cell companies. In order for solar cell companies to grow quickly enough to help deal with global warming, they would need profit margins similar to Intel or Apple. but fierce in-industry competition limits their profit margins to near zero. Gallium arsenide is a material that doesn't necessarily have the same capital bottlenecks as silicon. This is because gallium arsenide absorbs light 100 times more effectively than silicon, and so a gallium arsenide solar cell only needs 1/100th of the material. Because half of the capital cost of making a solar panel comes from turning dirt into a semiconductor, a gallium arsenide solar panel can be half as capital intensive as a silicon solar panel. Gallium arsenide solar cells are already mass produced. They're used on satellites because they're more efficient than silicon, and they're also radiation hard. Unfortunately, the traditional processes for making a gallium arsenide solar cell require extremely expensive equipment and materials that make it unviable for everyday power generation. However, there have recently been two advances that make terrestrial utility scale gallium arsenide solar cells viable. The first, developed by the National Renewable Energy Lab, is a technique for turning gallium arsenide ingots into extremely thin films that are perfect for solar cells. The second, and what I've been working on, is a low-cost, high-throughput way for turning gallium arsenide into solar cells that are performance competitive with commercial silicon solar cells. We expect that by combining the NREL's advances in making gallium arsenide films with my process for making gallium arsenide solar cells, we can make solar panels for half the price of both capital and per watt compared to commercial silicon solar cells. Despite these advances, though, any hypothetical gallium arsenide solar cell company will only be profitable until competitors start to enter the market. And so, at best, what my work will do is reduce the cost of building the factories necessary for us to effectively mitigate global warming. Thank you for your time.
Thank you. That was Philip Chehelka. Phil is a graduate student in the Atwater Lab. His overall goal is to develop low-cost solar cells with a focus on scalable manufacturing. For his current project, he is fabricating solar cells for the Space Solar Power Project to beam power from solar arrays in orbit to the ground. Next up is Andrew Hill from BBE, David Prober's lab, understanding how melatonin promotes sleep using zebrafish.
I'm a graduate student in the Prober lab where we use zebrafish larvae to study sleep. I think sleep is one of the most interesting biological processes because we all sleep every night, give or take, and yet there's still so much that we don't know about it. So my thesis focuses on how one particular molecule, melatonin, regulates sleep. You've all probably heard of melatonin. It's available over the counter as a sleep aid, and most people have some first or secondhand story about its effectiveness. In fact, since its discovery, there have been a number of conflicting reports that have led to a lot of doubts and controversies about whether or not melatonin is actually sleep promoting. We make melatonins in our own brain in a structure called the pineal gland, and its synthesis is in rhythm with the 24-hour clock, with a peak of melatonin production at night. Now, if you want to know if a molecule is important for a process, the best thing to do is remove that molecule and then ask if that process is effective. So the Prober lab removed melatonin from zebrafish by deleting the gene responsible for its synthesis. These fish don't make melatonin, and they slept about half as much as normal fish. So this suggests that melatonin made in the zebrafish's own brain is essential for nightly sleep. Now, I wanted to follow up on this and use zebrafish to understand how melatonin is promoting sleep, and I tried to do this in a couple of ways. First, molecules like melatonin usually work by binding a receptor, and there are two melatonin receptors. They're called MT1 and MT2, and it's totally unknown in any animal if one or both of these receptors is required for sleep. So to figure this out, like I said before, I need to remove these receptors and then look for changes in sleep. So I found that MT1 mutants were strongly impaired for sleep, just like animals that don't make melatonin, and I did not see this effect in the MT2 mutants, because they slept normally. Additionally, when I give melatonin, MT1 mutants they failed to fall asleep. That is, they were resistant to melatonin. So these experiments suggest that melatonin is acting through the MT1 receptor to promote sleep and not the MT2 receptor. Now that I know what the relevant receptor is, I wanted to know where in the brain melatonin is acting, and this will give us a clue as to how it promotes sleep. I found that melatonin receptors were expressed in brain areas that also have to do with visual processing. So this led me to hypothesize that maybe melatonin promotes sleep, at least in part by turning down our brain's responses to light and visual input. When we sleep at night, we have what's called an increased arousal threshold. This means that we're less likely to respond to stimuli that normally we would respond to when we're awake. This is a hallmark of sleep, but it's not really known how the brain does this. I think melatonin is playing a key role in this, which is really exciting, and I'm currently developing assays to test this hypothesis. Through my findings, I hope to shed new light on how this popular yet controversial molecule regulates sleep. Thank you.
Thank you, Andrew. Andrew Hill is a graduate student in the biology PhD program. Before coming to Caltech, Andrew studied sleep at Cal State University Northridge using the nematode worm C. elegans. He wanted to continue studying sleep using a more complicated organism and chose to investigate sleep using zebrafish in David Probert's lab. He is currently wondering which animal he should use to study sleep next. Thank you. Benjamin Hagi, EAS, Azita Imami in the MICS lab, robust learning systems for biomedical applications.
Hi everyone, I'm Eric Legrand. I'm 32 years old. Some of you may know me. I'm the former American football player in Rogers College. It was always my dream to become a football player since my childhood. In 2010, finally this dream became true when I was among the best defensive tackles in the United States. But it was just before the end of my career as a football player in the same year. I remember it was a game between Rogers and Army that I just put my head down and started running toward the shoulder of the ball holder. We got a point for that tackle, but I just fell down on the ground for several minutes and I had lots of pains. It was like a 1,000-pound block is pushing me down. I saw people around me and they took me to the closest hospital, where doctors told me that I became paralyzed down from the neck. Then after that, I were moved to the Kessler Rehabilitation Center. I just started talking on behalf of a spinal cord injury patient. Do you know that there are about 250,000 spinal cord injury cases all around the world with a treatment cost of about $10 billion? And most of them ask themselves that, is it possible that one day we can have normal life again? And usually they don't get any answer. Today I'm here to answer their questions with a positive answer. And this is where my PhD thesis starts. Brain-machine interfaces. Brain-machine interfaces has the potential to improve the quality of life for spinal cord injury patients. Although there are some BMI systems with high performance, they cannot maintain their performance for a long time. In my PhD, I'm trying to design intelligent systems as well as the corresponding hardware for them to answer three main questions. The first one is that what approach should we use to maximize the information which is extracted from the neural activity inside the brain? To answer these questions, we recently designed FINET, a feature extraction neural network, which is able to find an optimal solution to maximize the information, and it had outperformed the previous traditional techniques. The second question is, what decoder should we use to decode this neural activity to body movement? To answer this question, we designed a DRNN, a dynamic recurrent neural network, which is able to model the complex relationships in the brain. And the third part is that how can we translate these algorithms to a product? Now we are exploring efficient hardware solutions that are low power as well as small area. I hope one day we have enough information about the brain such that we can improve the quality of life for these spinal cord injury patients. Thank you.
Thank you. That was Benjamin Hagi, a PhD candidate in the Department of Electrical Engineering at Caltech. During his PhD at MICS lab, Meeks lab, under the supervision of Professor Azita Imami, his research focuses on the development of robust and high-performance closed-loop learning-based algorithms for different applications, ranging from biomedical applications, such as brain-machine interfaces, and ECG arrhythmia classification to autonomous unmanned aerial systems, UAS control. He also designs and implements the circuits, technologies that comprise the efficient developed algorithms. Very cool. Namita Saraf is from BBE, Lulu Chen's lab, and her talk is a maze-solving DNA robot.
When you think, oh, sorry. Okay. When you think of a complex robot, you probably picture something like Centauro, which is an emergency response robot. It's about 5 feet tall, it's made of metal, and it accomplishes the high-level task of emergency response by coordinating simpler tasks, such as walking, picking up debris, breaking down doors, and so on. It's able to interact with and manipulate its physical world. In our research, we're interested in a similar goal, but at a much smaller scale. So rather than being made of metal and manipulating debris and doors, our robots are made of synthetic DNA and manipulate small molecules and nanomaterials. So some tasks that our robots might be suited for are coordinated assembly of a tissue or allergen removal from food. Similar to Centauro, in order to achieve these higher level functionalities, our robots need to be able to develop simpler tasks that they're able to put together. But before we talk more about what these robots are able to do, let's talk about how they're built. So DNA is a material that's as interesting for its chemical properties as for its biological ones. It has very strong and very specific chemical bonds. So what you might remember from grade school biology class, A's and T's bind together and C's and G's bind together. So we can take advantage of this property to design custom DNA sequences that when we mix them together, create specific physical systems. We can also take advantage of thermodynamics to build dynamics into these systems without needing to use an external power source. So now let's talk a little bit about what these robots might be able to do. So what could a nanoscale robot that can interact with its physical world do that an electronic computer can't? So imagine that you have a severe allergy and a plate of food in front of you that contains that allergen. What if you could deploy your robot to get rid of that molecule? So in order to achieve this task, the robot would need to perform several simpler tasks. It would need to be able to navigate the complex environment of the food. It would need to be able to pick up the molecule, and it would need to be able to retrace its steps to get back out of the environment. So before we can develop a robot that does that, we need to make sure that these simpler tools are in our toolbox. So my research focuses on developing one of these skills, which is navigating a complex environment, or maze solving. Similar to how you might design an algorithm for a computer to use to solve mazes, we've designed an algorithm that a robot can use to solve mazes. When the robot moves forward, it modifies the track in order to record its progress. When it moves backward, it removes that modification so that if it hits a dead end, it doesn't record the wrong track. When it reaches the end of the maze, it will have developed a trail that is equivalent to the solution of the maze. In my field of molecular programming, we want our physical world to be as programmable as the world inside your computer. DNA robots are one way we can achieve that. As we build more sophisticated systems, we'll be able to compose them together to manipulate the world in more interesting and useful ways. Stay tuned.
Thank you. Thank you, Namita Saraf. Namita is a graduate student in Lulu Chen's lab studying molecular robots and information-driven nanoscale self-assembly. She is involved in Women in BBE and Caltech Letters, among other organizations on campus. Finally, Gregory Roberts from EAS, Andre Ferron, Nanoscale and Quantum Optics Lab, recovering the lost light in your color camera.
You're wrapping up an afternoon at the beach with the breathtaking sunset. It's starting to get a little dark, so you take out your phone to memorialize yourself set against the fiery, glowing sky. Later, you look back at your photo and realize that in the dim foreground, you appear grainy and dull. How is this still the case after so many years of mobile camera technology development? Your color camera is surprisingly inefficient and loses almost 70% of the light coming into it. When you take a picture, the scene is projected by a lens onto an array of sensors. These sensors are grouped into sets of 4, each one responsible for recording how much of a specific color is present at this point in the image. Those colors are categorized by bands of wavelengths. Red contains the longest wavelengths and blue the shortest. Think of red like a basketball, green like a tennis ball, and blue a ping pong ball. Each sensor has a filter on top of it, which only lets through certain wavelengths by checking what size they are. All the balls that are the wrong size are thrown away, meaning this filter discards 2/3 of the light that hits it. When there isn't a lot of light around, at dusk for example, lowering the signal in this way makes for noisy and grainy pictures. What if instead of rejecting the wrong wavelengths, we redirected them to the correct neighboring sensor in the group? Then, each location would still monitor a specific color, but no light would be wasted in the process. When a beam of light hits a material like glass, it can change direction. A curved lens, for example, bends light to focus it, but mostly acts the same on different wavelengths. It turns out if we carve tiny patterns in that glass at resolutions finer than the smallest wavelength, the varying size balls of light that hit it bounce around and shoot out in different directions. By simulating how light interacts with these complex shapes, we can search for and ultimately design patterns that send each wavelength to a precise location. Some can pass directly through, while others are routed to the appropriate neighbor. Once we know what pattern to etch into the glass, the problem becomes how to make something so complicated and small. The device I described is the size of a camera pixel, with patterns 10 times finer than its width. Fortunately, techniques to fabricate devices like this drive one of the largest industries. The computer chip in your laptop is made of a dense electronic circuit, wired together through a three-dimensional network of metal connections with widths small enough to generate these intricate patterns. By replacing those metal connections with glass, we can make cameras that are 3 times more efficient. This means higher quality pictures, better low light and high speed imaging, and sustained performance at ever-increasing camera resolution. Thank you.
Thank you. That was Gregory Roberts. Greg grew up in Pennsylvania in a family of 4 siblings and came to California for undergraduate studies at Berkeley, where he studied electrical engineering and computer science. After graduation, he worked for a couple of years as a software engineer for an augmented reality startup before deciding to come back to graduate school to study physics. He is currently a 5th-year student, Andre Ferron's nanoscale and quantum optics lab in the Applied Physics Department. His research focuses on designing new kinds of optical components and verifying them experimentally. Outside the lab, he enjoys running, triathlons, soccer, and hiking. Thank you so much. All right, now we're going to go into the next phase of the evening here.
Yes. So I'm pretty excited right now for the amazing presentations that we just had. Please join me in another round of applause for all of our presenters. So now we enter the judging phase of our event this evening. And while our judges are making the last of their notes and we're tallying up everyone's scores, Brian and I are going to tell you a little bit about some of the things that have been going on around the library and also around the Caltech Theater. And we have a very special guest that's going to be joining us for this evening. So I'll start and tell you a little bit about what we've been doing in the library this year. There are a lot of things that have been going on behind the scenes that you may not have noticed. We actually implemented a brand new library services platform. So if you go to search the Caltech library, you'll have noticed that the search interface has changed, but really everything that powers our library has changed. We moved to a new tool called Folio, and we're working with EBSCO, which is one of our partners for the event this evening on that. We're also in the midst of redesigning our website. And we've got a couple of new spaces on campus that you may or may not have visited yet. Creating more outdoor study space has been important for us in this time of COVID. So in addition to the SFL amphitheater space, we now have the Dabney Lounge, which is the area just outside of the Dabney Library. It has some study tables and some comfy chairs for people. And we also have the Tech Hub, which is the first floor of Caltech Hall. It's home to the Tech Lab, our 3D printing and fabrication lab, but also to our equipment lending service. So those of you who have iPads and laptops checked out, that's where you're going to return those. And we also, if you've been in there, have established a partnership with Vroman's, the famous Pasadena independent bookstore, to provide Vroman's at Caltech, which is a collection of actual science books that you can browse on campus and order from Vroman's. You can also have anything that's available via the Vroman's website delivered to campus through that service. We've also been working to rebrand the Sherman Fairchild Library, affectionately known as SFL, as the stress-free library during midterms and finals. So we've done a few fun things like invite the therapy dogs from the Huntington Hospital's therapy dog program over during finals and midterms. We also celebrated Einstein's birthday and Pi Day. We still have individually wrapped moon pies at SFL, folks, if you are interested. And we'll be doing it all again in another couple of weeks as midterms for spring start up again. So it's never a dull moment around the library. We hope that in addition to supporting everyone's research and data needs on campus, that we're also providing a welcoming environment for those of you who are looking to study, to write your thesis, and all that sort of thing. And now I want Brian to tell us about what's going on in the Caltech Theater. You have a show right now, right?
We do. Yes, we have a Thursday night, Friday night, Saturday night. We have an adaptation of a Jules Verne novel called From the Earth to the Moon. It is a big musical with 20 actors, undergrads, grads, and JPLers. It's $5 for Caltech students. So come see it. We've got 4 more shows. It's really fun. It's upbeat. It's really positive. It's a happy thing. So I think we need a lot of happy right now. So I'm really excited to wrap. We've been working on it for like 7 months right now. So we're like in that glow before the end, you know. The other thing, I just want to give a shout out to Professors DeBeery, McKeon, and Greer for helping to institute a storytelling module that's happening in the Engineering Applied Science program. It's called Credible Messenger Training, which is a collaboration with JPL and the EAS to help find ways to sort of normalize storytelling in many ways, kind of socialize this idea of what you all are doing and how important it is right now to be able to tell science back to a skeptical public. And I think my own personal opinion is that scientists are needed more now than ever of so many things that are happening in the world right now. We need scientists, that repository of truth, that you all get out there and be able to tell your science in a relatable, credible way, and to help save the world in many ways, too, with all the research you're doing. I also want to give a shout out to Ilana Smith and Barbara Avwak over in the International Scholar Services. They are helping to support a mentorship, another storytelling module with postdocs, which we've been doing for the last 10 weeks, which is really exciting to offer opportunities for postdocs to kind of navigate their lives here at Caltech. So just love that. And also, if you want to become part of our community of practice, many of us, Suzanne Hall and Robin Javier, Monique Thomas and Kara, and a lot of folks up at JPL are really trying to build this bridge of normalizing the need to tell stories and to be with each other and develop this community of storytellers in ways that connect with the public, but with each other too, and making it a more welcoming and comforting environment, that we all are together in this struggle to communicate our science. And so, yeah, that's all I got.
That's pretty amazing, Brian. Thank you very much for sharing that with us. So now it's time to ask you to take on the role of judges, and we're going to kick off our People's Choice Award voting. We are running our People's Choice Award voting for both the folks here in the Hamitman Auditorium and also those of you who are joining us via live stream. The winner is going to be chosen by you, the audience, and the winner is going to receive $500. And audience, I need you to please get ready to vote. Each of our presenters this evening has a code, and to vote, you text the code of your favorite presenter to the number 37607. For those of us who are here in the audience, you should be able to see on the screen, as well as those of you who are on the live screen, everybody's code. And while all of you are voting, I would like to invite last year's 3MT prize winner up to talk to you all, Mia de los Reyes.
Hi everyone, I hope I haven't been overhyped as the very special guest. But first of all, I just want to say a huge congratulations to all of the amazing presenters today. I can tell you from personal experience, it is really freaking difficult to fit years of research into 3 minutes, and I think all of our presenters did really amazing jobs, so. I think their jobs have also been made more difficult because they had to present live. So last year, because of COVID, I actually ended up presenting over Zoom, which was great because I was sweating like a maniac, and so I had these massive sweat stains, but no one could see it. So huge congrats again for managing to hide all of your nervousness, because I think you all did that very well. So I'm really excited to have heard so many great things. I think last year was tough for me. When I presented at the 3-minute thesis competition, I was not in a good place with my PhD thesis. And so presenting it to other people actually made me realize that I actually like what I do, and that it's kind of cool. And also that I really like talking with people about it and presenting. And it was really good practice for job talks and for, that kind of future thing. I don't know if there was anything else I was supposed to talk about. I think I'm just supposed to, buy us some time to finish tallying votes and for the audience members to continue voting. So thanks again, and congrats again to all of our amazing presenters. You all, no matter what the outcome is with the results, I thought you all did an amazing job. I've learned more in the last 14 times 3 minutes than I have in, honestly, a while. The 3-minute format has been really great for my Zoom-fatigued brain, which now doesn't have much of an attention span. So I think you all did really fantastic jobs. You guys are all great. Thanks.
Mia's here partially to show you all that there's life on the other side of 3MT. And that you can use what you learned here to actually help propel you forward in your science communication. I'd like to give some special shout outs to our communications coaches for this year's 3MT competition. All of our finalists were offered the opportunity to partake of an optional communication coaching session, and I would really like to thank those communications coaches for the generosity of their time and the help that they were able to give the finalists who took them up on their coaching. So I'd like to say thank you to Chris Daly. She's an HSS instructor in writing, and she also happens to be the library's communications coordinator. I'd also like to say thanks to Paul Propster, who's the story architect and senior communications strategist at NASA and JPL. And a special shout out to the staff at the Hickson Writing Center: Leslie Rico, Noreen Manukian, Josh Schneiderman, Aaron Burkett, and Bridget Alex. And I'm looking at the door because I don't think that we yet have the judging tallies. So I have a couple of other thanks that I would like to add, if you all will indulge me. I would like to say a special thanks tonight to our Caltech Library operations team: Jocelyn Yamasaki, Catherine Geard, and Matt Buga. They made all of the things that you see tonight happen. I would also like to thank several other folks who have made this event work tonight. I'd like to thank, from the library, Melissa Ray, Ling Lim, Bianca Rios, and Penny Nader Muro. I'd also like to give a special shout out to the grad office and Jacob Dalton for the help tonight. Do you have it? I'm being whispered to that we have results from the People's Choice Award. We've only done this one time before, so.
Oh, dang it. we'll just go back to the slideshow. Sorry.
Not yet?
Can you know that?
So while we're figuring out how to get the poll everywhere, I was wondering, Brian, if you would tell us a little bit about your storytelling class that you teach every term here at Caltech.
Yes. Yeah, come if you want to just come and play a little bit and get comfortable being in front of people and developing the confidence needed to stand in front of people and convince them to give you money for more research and things like that. For sure, come. I think the biggest thing for me is breathing. When people are speaking, I find that to be one of the things that grounds people the most. Like when we get really excited and our temperature is rising and the blood pressure is like just pounding. Being able to kind of relax in front of people and to just trust that you're going to connect with others. And that for me is, I think, one of the big things of how do we connect our science with what people care about? Like, how do you connect, something's happening here. How do you connect with your audience in ways that make us care about your science, right? There's a certain amount of empathy that happens when we're listening to science that moves us. And I see Kara being moved a little bit right now with some excitement.
I might have the results on this piece of paper. Have you figured out the People's Choice yet?
Yes, but I will click the present button until you tell me.
Okay, so we're actually, now that we figured out how to do the People's Choice Award, we're actually going to save that one for last. And I will make the announcements of the first, second, and third place awards, which we have these lovely 3D printed trophies for them. So if you don't mind, when I make your announcement, would you please come up here and collect your trophy? And our photographer right back there will take your picture. So number 3 for the third place award, I am delighted to present to Skylar Ware, Sports, Drinks, and Batteries, and Beyond. All right, and our second place winner is Emily DeYoung, Clouds Microscale Physics with global impact. And now, what you've all been waiting for. The first place award. Yes, drum roll, please, Catherine. It goes to, you ready? Planets on a Galactic Scale, Ida Damars. All right, and the moment we've all been waiting for, the People's Choice Award goes to Skylar. Well, all right, everyone. I've had a wonderful time tonight, and I would like to say a special thank you to all of tonight's participants, our esteemed judges, and to our very generous sponsors. And thank all of you for attending this evening. We look forward to seeing you at next year's 3MT competition. And for those of you who are joining us in person tonight, we have some light refreshments outside on the Cahill patio. Thank you.
Thank you. Yay. Yay 3MT. Yay library.